So now that we've talked about how to make gametes, we can now talk about how gametes come together to create an embryo through the process of fertilization. So fertilization begins with sperm reaching the egg, but as we discussed, there are lots and lots of sperm and only one egg. Ultimately, out of the millions that are released with each ejaculation, only about a couple hundred are going to reach the egg. Now, capacitated sperm, those ones where the acrosome is prepared to release enzymes that will ultimately allow them to get into where the oocyte is, those capacitated sperm can react to chemical factors that the egg is releasing, and that sort of draws them in, guides them to where the egg is. Now, the first step for a sperm who has reached the egg is to get through the follicle, okay, that surrounds the oocyte, and then when it reaches to the zona pellucida, there are factors there that allow it to bind to the surface of the zona pellucida, and that binding is actually species specific, which is the reason that a horse sperm can't fertilize a cow egg. But once the sperm has bound to the zona pellucida, the acrosome then releases those enzymes, which allow the sperm to burrow into the zona pellucida, degrading the zona pellucida, and then to enter into where the oocyte is. Once the sperm is into where the oocyte is, in through the zona pellucida, the sperm cell membrane and the oocyte cell membrane will fuse. Now, that fusion of the two cell membranes triggers a response designed to prevent polyspermy, two sperm fertilizing one egg. So what you're seeing in the video here is on the right-hand side is a wave of calcium that sort of rushes across the oocyte. And then cortical granules beneath the cell membrane of the oocyte are going to release their contents. And the sperm receptor molecules on the zona pellucida, the one that initial sperm had bound to, will no longer accept new sperm. So that process takes care of the sperm that still have yet to bind to the zona pellucida and those that were in the process of burrowing through the zona pellucida, making it impossible for additional sperm to meet and fuse with the oocyte cell membrane. And that's important because if two sperm fertilize one egg, what you end up with is triploidy. And in fact, dispermy is the most common cause of triploidy. So after fertilization, once the cell membranes have fused, the oocyte can now return to meiosis, which of course it had stopped many years before. It produces a second polar body, and interestingly, the first polar body goes through another round of meiosis too, and so we end up with three polar bodies altogether, which is what's shown here. But the second polar body is produced, and now the oocyte is called a definitive oocyte. And a fertilized oocyte is now called a zygote. The whole package together is called a zygote. Ultimately, the male and female pronuclei will fuse so that we have a single-celled embryo with a single nucleus, and now we are ready to start talking about how that makes a baby. The first step in that process is cleavage. And cleavage is interesting because now the embryo is going to go through a series of mitosis, so a series of cell divisions that are mitotic, not meiosis. We're done with meiosis. But we're not going to see any growth. So the original fertilized egg, now called a zygote, becomes many smaller daughter cells that are called blastomeres, okay? But we don't change the overall size of the embryo. And it's still surrounded by the zona pellucida. But after 4 days, the embryo will have anywhere from 16 to 32 cells, and now you call it a morula. And that comes from the Latin word morum for raspberry, and it does look a little bit like a raspberry. Now, once a zygote reaches the 8-cell stage, the embryo starts to develop a polarity, so it starts to have an inside and an outside. Before it was just a ball of cells, okay? And that process starts when the blastomeres start to try and maximize the contact that they have with the blastomere next to them, okay? Also, some of the blastomeres are going to start to move towards the interior of the embryo, while others will move towards the exterior. So the ones that move on the inside become the inner cell mass, and that's going to become the embryo. The ones that move towards the outside become the trophoblast, and that's going to become the placenta, ultimately. And it's actually the expression of transcription factors that determines which cells are going to move in which direction. certain transcription factors are expressed in certain regions of the embryo and not in others, creating this sort of pattern. Now, after that, the morula is going to develop into a blastocyst, okay? And that involves creating a certain level of polarity within the embryo. Now that starts with the trophoblast. So the trophoblast cells begin to adhere very tightly to each other, and ultimately we want them to become watertight. They do that through a number of mechanisms. They express E-cadherin, which is an adhesion molecule on their cell surface. They form intracellular junctions, and the trophoblast cells begin to express an ion channel. So now that they've sort of become watertight and they're expressing an ion gradient across this trophoblast membrane that we've created, now we get water osmosing in to the middle of the embryo, and that's about 4 days post-fertilization, and that forms a cavity called the blastocele. At the same time, that inner cell mass, those blastomeres that move to the interior of the embryo, are compacting and sort of forming a clump on one side of the embryo. So now the embryo actually has a side. It has an embryonic pole, which is the side with the inner cell mass, and an aembryonic pole, which is opposite the side with the inner cell mass. Once the embryo has become a blastocyst, it's now ready for implantation. And that process starts with hatching. So we don't really think of human eggs and embryos as hatching, but they do. They have to escape the zona pellucida, and that's what's being shown in the video here. Once the blastocyst is outside of the zona pellucida, it's now able to attach to the uterine lining. And the adhesion of the blastocyst to the wall of the uterus triggers the uterus to sort of vascularize. Blood vessels grow into that space to support the uterine lining and the developing embryo. We also get this hormonal feedback loop going where the corpus luteum, so the former sort of follicular structure that was supporting the oocyte in the ovary, is secreting progesterone, and that helps maintain the uterine lining so it doesn't slough off like a period. But after implantation, the trophoblast cells from the blastocyst are going to start making HCG, which is human chorionic gonadotropin, the pregnancy hormone. And that prolongs the life of the corpus luteum. So then we get more progesterone, which supports the embryo further. Now, by 11 to 12 weeks, the corpus luteum is going to degrade, and the placenta will take over making progesterone to support the uterine lining. The second embryonic week, we get more progress with implantation. So between days 6 through 9, the trophoblast now starts to differentiate. Before, the cells were sort of all the same, doing the same things, just in different places. But now they're starting to do different things. So the cells of the trophoblast that were sort of up next to the endometrium, the uterine lining, they start to lose their membranes, and they merge together to form this sort of large mass of former cells with lots of nuclei in them called the syncytial trophoblast. The cells that line the blastocyst, the trophoblast cells that are sort of adjacent to the blastocyst itself, become the cytotrophoblast. Okay. Now the cytotrophoblast is going to release enzymes that degrade the extracellular matrix between the endometrial cells. So it's essentially degrading the lining of the uterus. At the same time, the syncytial trophoblast is going to extend itself. It's going to grow into that space that the cytotrophoblast created by degrading the endometrial cells. And as the syncytial trophoblast grows in to the endometrium, it pulls the blastocyst inside with it. And so by day 9, the embryo is fully implanted within the uterine lining. Now it's interesting to note that implantation almost always occurs in the uterus, but not always. And when it doesn't occur in the uterus, it causes a serious problem for the mother and for the baby. So in an ectopic pregnancy, the blastocyst implants within a fallopian tube. That's really very dangerous for the mother because the embryo will very quickly grow to a point where it can no longer be contained within the fallopian tube, and the fallopian tube will rupture if the pregnancy isn't ended before that. Now, usually there's a lot of pain and cramping associated with an ectopic pregnancy, So the mother will usually notice before we get to the point where there is danger of rupture, but not always. And so if an ectopic pregnancy is recognized before rupture, then they either do surgery to remove it or they'll give methotrexate, which will cause the pregnancy to end on its own. But a lot of times you get scarring within the tube that impairs fertility down the road. If you have to do surgery, of course, you've lost the tube, which impairs fertility. You can, however, even have an extrauterine pregnancy. So a pregnancy that occurs, a blastocyst that implants outside the female reproductive structures altogether, on the outside of the uterus, on the membranes that line other organs in the abdominal cavity. That is extremely high risk. Clearly, those are not areas that are set up to support a growing baby, although on occasion you get reports of surviving infants, and if you watch enough TLC, you'll probably see